So far, we've been talking about meiosis and all the different steps. Now we're going to talk about what happens when things don't go as planned. But before we do that, let's just review how normal meiosis occurs in terms of what happens to the chromosomes. So we're going to skip the prophase stage and we're going to start with metaphase. So with metaphase 1, you see that we have a cell with two pairs of homologous chromosomes. And as we learned before, during metaphase 1, homologous chromosomes line up together on the metaphase plate. And then as a result of the first stage of meiosis, we separate homologous chromosomes from one another. So in this case, we're seeing these two blue chromosomes are going this way and the red counterparts go to the other cell. And then as a result of metaphase 2 and the whole process of the second stage of meiosis, we're going to get 4 gametes, each of which have one of the chromosomes in them. Okay? But what happens if separation of homologous chromosomes during the first meiotic stage, or the separation of sister chromatids during the second meiosis doesn't happen? When chromosomes don't separate when they are supposed to, we call that process non-disjunction, when separation fails. So let's take a look at the first scenario where the non-disjunction happens during the first meiotic division. So this is the homologous chromosomes lining up at the metaphase plate, and during anaphase they're supposed to separate, but look, the small chromosomes separate, but these big ones fail to separate. So homologous chromosomes do not let go of each other. So as a result of the second meiosis, where the sister chromatids separate, and then you're going to get 4 cells. 2 cells are going to have 1 extra chromosome. So they have 2 long chromosomes and 1 short. This situation we call an n+1, because if these were to separate properly, each cell would have n chromosomes, which is consistent with the n number for that organism. In this case, the n number is 2, but we have 2 plus 1. So we're going to get 2 gametes that are n+1, and then the other 2 gametes that come from the other cell, they're missing the long chromosome. So they are n+1, in this case 2 minus 1. So that's when non-disjunction doesn't happen during the first meiotic division. Non-disjunction can also happen during the second meiotic division. So the homologous chromosomes separate properly, but during meiosis 2, sister chromatids don't separate. So in this case, the blue large chromosome, the sister chromatids separate, but the little one, for the little chromosome, in our example, the sister chromatids don't separate. So from this cell, in which sister chromatids didn't separate, we're going to get 2 different types of cells. One is n+1, which has 2 little chromosomes and 1 big one. In this one, the little chromosome is missing, so it's n-1. In the other cell where the separation of sister chromatids happened properly, we just have 2 normal cells which have n number of chromosomes, which in this case is 2. So now, let's assume that these are egg cells, okay? What happens when these egg cells are fertilized by a normal sperm? In this case, if a normal sperm with n number of chromosomes fertilizes the cells with n+1, you're going to get a cell that has 3 long chromosomes and 2 short. So we got an extra chromosome. And what is the extra coming from? We have 3 of the same kind of chromosome. We call this trisomy. Tri stands for 3. So this cell has trisomy in it. The other cells which are lacking the long chromosome, they're going to be fertilized by a normal sperm, and you're going to have 2 small ones and 1 large one. So this is the, if you haven't figured it out yet, this is the result of the fertilization. So that's the zygote. So in this case, we have 1 long and 2 short, where we're supposed to have 2 long and 2 short, right? So we call this individual with a condition of monosomy. Mono stands for 1. So you only have 1 of the 2 homologous chromosomes. In this case, we have the red one, but we don't have the blue one. On the bottom, when we are looking at non-disjunction during meiosis 2, if, again, this N+1 is fertilized by normal sperm, then we have 2 long and 3 short. Again, we have trisomy. In this case, the trisomy is with a short chromosome. And in the bottom stage, it's a normal egg fertilized by a normal chromosome. So you're going to get 2 long chromosomes and 2 short chromosomes, which is a diploid. Okay? And that's normal. So in these cases where you have an uneven number of chromosomes, we say that they are aneuploid. And aneuploidy is an abnormal stage. This is not normal. But when you have the correct number of chromosomes, 2 at each chromosome, you have a euploid individual, which is a normal situation. Now you may say, well, these are just exercises on paper, but actually things like this happen. We're going to see a human example of it, but non-disjunction actually happens quite a lot in plants, and that's one of the ways that actually plants evolve. One example of a human condition that is a result of non-disjunction is Down syndrome. And Down syndrome is a result of non-disjunction, where at some point the either homologous chromosome 21s or the sister chromatids don't separate, and the individual that is born has 3 chromosome 21s. Now, the occurrence of Down syndrome happens, it has a direct correlation with the mother's age. And after the age of 40, the occurrences of Down syndrome goes way up, and it is thought that since females are born with their eggs already all made, and females' eggs are arrested after the first meiotic division. So they don't complete the entire meiotic stage, and every month when an egg is released in a mature woman's body, then that egg undergoes the second division. And it is thought that because the eggs are present in a woman's body, and maybe we exactly don't know why, but one of the thinking is that since the eggs are in a woman's body and we are during our lifetime, we are exposed to so many environmental conditions, or maybe if the eggs lie around too long, they accumulate abnormalities. Anyway, it is thought that it has to do with the age of the egg that increases this condition of trisomy. Now, you can have trisomy of sex chromosomes as well. So chromosome 21 is a non-sex chromosome, but we have situations, real life situations, where there is a sex chromosome abnormalities, and we have a number of them here. So for example, if the individual has 2 X chromosomes and a Y, the individual is male. Now, every time an individual has a Y chromosome, it will develop as a male because the male determining gene is sitting on the Y chromosome. Now, this situation the syndrome is called Klinefelter syndrome. There is an extra X chromosome, there's less facial hair or muscle mass, and it is thought that the origin of non-disjunction is in the egg or sperm, and the frequency is 1 in 1,000 live male births. A second situation is XYY. there is really no syndrome associated with it, and it is thought that it's an error in meiosis during sperm formation. So yes, errors also happen during sperm formation, and that's in 1 in 1,000 live male births. You can have situations where an individual which presents as a normal female, it has 3 X chromosomes. Instead of having 2X, it has 3X chromosomes, and the non-disjunction could happen in egg or sperm, and it happens in 1 in 1,000 live female births. And finally, we have individuals where they only have 1 sex chromosome. There is no, this O stands for nothing. So there's neither a second X chromosome or a Y chromosome. And because Y chromosome isn't there, the person presents as female, but they do have abnormalities, which is called Turner syndrome. And this error can happen because of non-disjunction in either sperm or egg. And it happens in 1 in 2,500 live female births. Now, you may be wondering, okay, why is it okay for an individual to have 3 X chromosomes? Well, this is because of natural occurrence in female bodies, what is called X inactivation. Even in a normal female where there's only 2 X chromosomes, one of them is always randomly deactivated. So that if you were going to look at cells of females, you would see a normal X chromosome, but then you would also see some really dense material that doesn't really look like anything. It's called bar bodies. Okay, so interesting manifestation of that is this in this tortoiseshell cats. The gene that determines the color of the fur in these cats sits on the X chromosome. Now, this entire cat has all of its cells have both of its X chromosomes, but randomly one of these X chromosomes will become inactivated. So in the parts of the cat where the orange color is presented, the X chromosome that carries the black gene for fur color is inactivated. In cells where there is black patches are being expressed, the X chromosome that has the orange allele or orange form of the gene is deactivated, and then the ones that is expressed is the one with the form of the gene that gives us black color. So that's an interesting manifestation of what happens in females. So what happens in X inactivation in triploid individual? Well, these two are not triploid. These are normal male and female. In females, one of the X chromosomes is inactivated. In males, no X chromosome is inactivated because there's only one. In XXY males, one of the X chromosomes is inactivated. And then in XXX females, 2 of the X chromosomes are inactivated. So that's why a female can have 3 X chromosomes and be fine. Now, non-disjunction is not the only kind of chromosomal abnormality. There are times where a part of a chromosome just gets deleted. This is basically called chromosome deletion. One of the real-life examples of such a situation is Cry-Duchette syndrome. And the reason this name is given to it, it's in French. That's because children that are carrying this problem, children that have this part of their chromosome 5 missing, they cry like a cat when they're babies. And then here's an example of a boy with Cry-Duchette syndrome at different stages of their life. They usually have mental disabilities. Other forms of chromosomal abnormality are inversion and reciprocal translocation. What is inversion? Inversion basically means flipping. So you have a chromosome. These genes are sitting on the chromosome, but for whatever reason, this part of the chromosome flips. And then you can Imagine that the order of the genes changes. Sometimes genes are sitting at the junctions of these flips and they get all screwed up. They don't work anymore. A common type of hemophilia is a result of inversion. Hemophilia is a form of disease when normally when we bleed, these clotting factors are going to stop the bleeding, but a protein that is involved in normal clotting of blood, factor VIII, is sitting in this region where the chromosome is flipped. The gene for protein factor VIII becomes inactivated, and therefore the individual develops hemophilia. That means every time they cut somewhere, they bleed and the bleeding doesn't stop, and this is a lethal condition. Another example is reciprocal translocation. This is when non-homologous chromosomes exchange parts. If you remember, during the prophase 1 of meiosis, we said homologous chromosomes exchange parts, and that's a normal occurrence, and it's a good thing. With reciprocal translocation, it's non-homologous chromosomes that are exchanging parts, and this is not normal. And a disease, a blood cancer, it's called chronic myelogenic leukemia, is caused by two chromosomes exchanging parts with one another. And these two chromosomes are chromosome 22 and 9. So why is this cancerous? Well, we talked about proto-oncogenes before. Proto-oncogenes are usually genes that are involved in promoting cell growth. That's their normal function, but they're normally regulated. The cell usually has a way of keeping them in check so they don't promote too much cell growth. Now, in chronic myelogenic leukemia, chromosomes 22 and 9 exchange parts, and a fusion of the gene ABL1, which is a proto-oncogene, is made with the gene BCR, and it's still, I don't think, clear what BCR does. And it doesn't really matter, because what happens as a result of this fusion is that ABL is no longer in a form that can be regulated. It turns into a form as a result of fusion with BCR that it's out of control. It promotes growth without any limitations. So it turns from a proto-oncogene into an oncogene. Now, it's really concerning when we can see that chromosomal abnormalities happen, and sometimes parents can look at the chromosomes of a fetus and they can check to see whether chromosomal abnormalities happen or not. Two of the ways that they can do it is by amniocentesis and chronic villus sampling. Basically, these involve taking samples of cells from a very early stage embryo, looking at their chromosomes under the microscope and looking for abnormalities. And there's a video that is linked here that's going to show us how.
Diagnostic testing in pregnancy provides patients with a specific diagnosis. Diagnostic testing is most often performed for patients who have an increased risk for a specific condition, possibly due to advanced maternal age, abnormal screening results, birth defects detected by ultrasound, or family history. Anyone can have diagnostic testing. Diagnostic testing requires the collection of tissue or cells from the pregnancy. This is more than a blood draw from mom. In the first trimester, the available invasive procedure is called a chorionic villus sampling, or CVS. This is a sampling of the chorionic villi, a type of placental tissue, usually done between 10 to 14 weeks gestation. This test is often done through the cervix, like a pap smear, though it can also be done with a needle through the abdomen into the uterus. In the second trimester, after 15 weeks gestation, an amniocentesis can be performed. An amniocentesis includes placing a needle into the abdomen into the sac of fluid that surrounds the baby. Fluid can be collected without touching the baby. In this fluid are fetal cells that can be used for diagnostic testing. Ultrasound is used during both the CVS and amniocentesis. These procedures are typically performed at specialized centers. Most women who have an invasive procedure have no complications. The risk for miscarriage when these procedures are performed by experienced providers is typically significantly less than 0.5%. Once the sample is collected, a number of different diagnostic tests can be performed. The type of testing performed depends on the reason a woman is having testing. The most common testing performed following a CVS or amnio is testing for chromosome abnormalities in the baby. Diagnostic testing provides a yes or no answer. Diagnostic testing is more complicated than screening testing and should be reviewed with a healthcare professional. If you wish to learn more about diagnostic testing, your physician may have you meet with a genetic counselor to discuss this option in more detail.